Dirk, thank you so much for the introduction. Um, I'm so happy to be here and talk in this lecture series. I'm especially interested on how pathology is manifested in language communication of patients with neurodegenerative conditions and stroke aphasia by developing tools and methods for natural language processing, acoustic analysis, and machine learning. Ultimately, my goal is to determine the relationship between brain and language. In this talk, I will be discussing how machine learning and natural language processing can be employed to study speech communication in patients with language impairments. I will play a few sounds and listen to the acoustic characteristics, the grammar of speech, and also the way speakers speak, like to identify characteristics of the speaker.
There um, was a picture of this kitchen with their mother, who is drying the plates and has an apron. There was a girl in looking reached for her cookies. The cookie jar, and his brother is trying to get to her and him, but they're doing it, he's gonna go tip off on the stool, and the mother didn't know what was happening when the head of the wall that the wall is going on in the wall onto the kitchen floor. And there's some these are curtains, and you can see something over on the other people, though, on that person, on that.
Let us listen to the second speaker.
Well, there's a kid that is trying to get this cookie jar out of this, but the thing is falling down and he's going to fall. And the woman who, like, should I say that's the mother? It's probably the mother. And she's in, shit, I can't think of the word of that, but there's water coming out of that onto the floor. That's screwed up, but she's washing stuff. I don't know if that's the right word. And then the daughter is, I don't know what she's doing to the brother.
And the third speaker.
Well, we're in somebody's home. They're in the kitchen. There's a mother. I'm going to say a son and a daughter. Mm-hmm. The mother's at the area where you wash the dishes. And the water is overflowing. And she's just standing there with the water going all over her feet. And she is wiping a dish. She has on an apron and little shoes, and her hair is somewhat short, and she has a sleeveless dress on.
There is a
well, we're in somebody's home.
Well,
they're in the kitchen.
There's a kid.
There's a mother.
So we can elicit a lot of information from these recordings. For example, we know the gender of the speakers. Okay, we know that these speakers are adults. We also know from just listening to the speech recording that these are speakers of American English. Um, so can we perceive if these speakers have a language disorder? We notice that they differ in speech production. For example, for the first female speaker, the first female speaker seemed to have articulatory problems. And the second speaker seemed to have problems recalling words, mostly nouns. The third had some difficulty and made recalling some words and made lots of pauses. These are patients with primary progressive aphasia, a neurodegenerative condition that affects speech and language. This talk centers around a question: can we use speech communication to inform diagnosis, prognosis, and inform and improve therapy decisions? So these patients have different PPA variants. I will discuss these variants later on in detail. But these variants basically indicate that they have a damage at different brain areas.
There is a
I will be arguing that speech communication can provide linguistic, interactional, and social markers that can enable the early identification of patients with dementia, such as primary progressive aphasia and Alzheimer's disease, identify patients with different symptomatology, and enable prognosis of neurological conditions. Specifically, speech communication provides information about the linguistic contributions of the interlocutors to the development of the macrostructure and microstructures of discourse, which include topic organization, information structure, and the cohesion of forms and coherence of meanings. We can also evaluate how interlocutors follow the conventions of speech and interaction to exchange turns, resolve overlaps, avoid interruptions, etc., using cues of turn completions and turn continuation. So as speech continuation takes place in dynamic sociolinguistic and multilingual environments, the successful conversational contributions are those that consider the identities of participants and the social, economic, etc. constructs that are associated with these participants. Other information we can elicit from just listening a part of speech conversation is non-linguistic information. For example, measures of language production that correlate with memory and attention, and measures that can indicate social characteristics, such as the variety or the accent of the speaker, and physiological properties, such as the age of the speaker, whether the speaker is an adult or a child, and whether the person is a man or a woman. And also emotional information, like being happy or sad. Most importantly, speech communication can provide information to identify the pathology of a patient. For example, in the recordings, the difficulties in grammar and articulation can help us to identify the PPA variant of the patient, which is a cue of the area of brain damage. So standardized pen and paper tests usually focus on specific linguistic domains, such as verb naming and object naming. The study of linguistic communication and speech and interaction can provide multimodal linguistic information and communicative markers that determine patients' linguistic, social, interactional symptoms due to neurodegeneration or stroke. However, Analyzing speech communication manually is a time-consuming and laborious process. For example, the time estimated to analyze manually a minute of discourse may require up to an hour to complete. So it is difficult for manual analysis to be administered frequently in the clinic, for instance, and to provide a more detailed picture of cognition changes over time. Clinicians must collect and transcribe language samples and annotate the linguistic characteristics they target. The variability of annotation schemas, annotation techniques, and the differences in expertise and training of clinicians in doing the annotation often makes the process even more difficult. Additionally, research is halted by lack of standardization. Here, I need to mention the great initiative by Brielle Stark and the Focus Aphasia group as they aim to describe best practices for analyzing discourse, reporting guidelines for discourse studies to facilitate the replicability of discourse analysis. Another problem is how to elicit speech communication in the clinic. Lucy Briand's Cestar talk dealt with discourse and discussed elicitation techniques using virtual reality and immersive reality, which can be employed along with more traditional elicitation approaches, such as map tasks and picture description tasks. Picture description tasks elicit connected speech, but these tasks impose constraints on the grammar and prosodic patterns that are elicited. Perceptual identification of speech characteristics just by listening is actually subjective and varies on how clinicians process, interpret, and judge acoustic, grammatical, etc. properties of speech and language. So, how can we analyze speech communication fast, elicit more information from it than we get from standardized pen and paper tests, and accurately? Automatic analysis of speech communication can provide objective measures and scores of language deficits. These can be elicited from the acoustic signal using signal processing or from text transcriptions using natural language processing algorithms. Moreover, automatic evaluations of speech communication can provide frequent assessments of language symptoms and provide a clear picture of the cognitive progression of a patient over time. So this talk has two aims: to identify early signs of neurological diseases based on acoustic and morphosyntactic measures of speech communication, and provide diagnostic tools for patients with dementia. So the overarching goal is to facilitate diagnosis, prognosis, and ultimately improve therapy decisions by providing less stressful, quick, and accurate evaluations. By the end of this talk, I hope I will have demonstrated examples of the contribution of machine learning and natural language processing in diagnosis and prognosis of conditions affecting speech communication. So signal processing, natural language processing, and machine learning can contribute to the diagnosis, prognosis, and ultimately for improving therapy decisions. Speech acoustics, for example, can provide substantial information about language pathology. For example, the relative vowel duration for polysyllabic word repetition task was found to distinguish patients with non-fluent PPA and logopenic primary progressive aphasia. Automatic morphological measures, such as the noun-verb ratio, can reveal whether an individual with PPA shows preference towards nouns or verbs. This information may here to subtype patients with the non-fluent PPA variant from patients with the semantic PPA variant. Um, so machine learning, acoustic analysis, and natural language processing are what I will be discussing here. The automatic acoustic feature extraction enable us to evaluate patients' acoustic productions and elicit information about the brain damage. Automatic morphosyntactic analysis provides information about how speakers with different pathologies produce language. And finally, machine learning enables us to employ the automatically extracted acoustic and morphosyntactic features and develop next generation diagnostic tools and tools for subtyping. We will discuss these steps one by one. I will start with differences of individuals with mild cognitive impairment from healthy controls based on their speech acoustics. As speech acoustics can be elicited easily from speech productions, it is possible to function as early diagnostic markers of mild cognitive impairment. For example, in a recent study, we analyzed recordings of the cookie theft picture description task produced by 26 individuals, 26 individuals from Sweden with mild cognitive impairment, and 29 healthy controls, and calculated measures of voice quality and speech fluency. Phonation and voice quality demonstrate how well patients control the sublaryngeal and laryngeal vocalization systems. So we measured the H1, the difference of the first harmonic H1 from the amplitude of the third formant, the A3. The stronger H1 indicates breathy voice. Okay. The sepstral peak prominence is a reliable measure of dysphonia and accounts for the periodicity in the voice signal. The center of gravity is, which is the average spectral frequency, can also distinguish patients with MCI from healthy controls. Shimmer indicates the variability of the amplitude from peak to peak, namely the local maxima, and indicates noisy productions and breathiness, as it is a correlate of glottal resistance. For speech fluency, we calculated the average syllable duration, the articulation rate, and speech rate. We found that patients with cognitive impairment differed significantly from healthy controls with respect to acoustic features of voice quality, namely the difference of the first harmonic and the amplitude of the third formant, shown in the top left figure. Patients with mild cognitive impairment had a lower sepstral peak prominence. This is the second plot at the bottom, at the top, an indication of greater dysphonia. And the center of gravity shown in the first figure, left to right from at the bottom. And Shimmer, which suggests that the speech of patients with mild cognitive impairment is noisier and with greater breathiness. They also perform worse than the healthy controls with respect to speech fluency, namely the articulation rate and the average speaking time, as the speech of patients with mild cognitive impairment is slower than the speech of healthy controls. This is also evidenced by the longer duration of syllables in patients with mild cognitive impairment. So acoustic measures can be employed to identify patients with mild cognitive impairment. Acoustic measures can also be employed to objectively score and compare treatment effects in patients with apraxia of speech in the non-fluent PPA variant. These patients received language therapy with transcranial direct current stimulation, and we want to estimate the effect of the transcranial direct current stimulation, TDCS, over the sham condition. Although I will be discussing a word repetition task now, a similar methodological approach can be employed to analyze articulation from conversational tasks. Specifically, we investigated whether transcranial direct current stimulation over the left inferior frontal gyrus coupled with speech intervention improves speech production more than sham with speech intervention. 11 patients with non-fluent PPA with apraxia or speech symptoms received either TDCS or sham, along with language treatment for 15 weekday sessions. Evaluations took place before intervention, immediately after, and 2 months post-intervention. Words were segmented semi-automatically. I will show you an example of this segmentation. Here we see a production.
Inservetolo.
So here we identify each vowel and consonant. As you can see, it is a pretty challenging task to segment the speech, even for humans. But here we used an algorithm that enabled us to do the basic identification of consonants and vowels, and then we did a manual correction. So what we observed is that TDCS combined with language results in significantly faster speech productions in these patients. The red lines in the figures indicate sham, the sham condition, whereas the green lines indicate the TDCS condition. The lower lines, that is the lines that are closer to the bottom of the figure, indicate an improvement. Namely, they indicate fast actions, shorter sounds. So the findings show that the effects of TDCS improved the speech productions and continued up to the post-intervention period in the trained items, but not in the untrained items. But overall, we can see that that TDCS has a substantial effect, a significant effect over sham by improving sound duration and resulting in faster speech productions in individuals with apraxia of speech. Sound duration extracted automatically from speech sounds shows the effects of treatment, TDCS versus sham, allows the comparison between different time points of treatment and conditions. Most importantly, as it is very difficult to score such fine changes perceptually in speech articulation, eliciting acoustic measures, durational, prosodic, spectral, can provide objective and accurate measures of apraxia of speech. So sounds can also provide social information. Automated measures from vowels and consonants can enable the identification of social properties of speakers. Speech sounds provide substantial information about the identity of the speaker. Listeners can identify, for instance, the gender and age, and also the accent or language variety of speakers, their dialects or sociolects. Therefore, it is possible to recover this information, and we can also recover social information, such as the dialect from speech communication, using acoustic measures and machine learning. And generalization of this approach can enable us to identify sociolinguistic information in patients with multidialectal and multilingual backgrounds. So vowel formants, vowel duration, and the fundamental frequency were calculated from segmented vowels in a study where I employed the C5 decision tree machine learning model to identify the dialect of speakers of two dialects of Greek. Then the table to the right shows measures of accuracy for the classification of dialect and stress, showing that the model was able to identify the dialect with 83% classification accuracy from just one vowel. It also identified whether the vowel is stressed or unstressed with 90% classification accuracy. The table to the left shows the ranking of the acoustic features from more important to less important for dialect classification. Similar results were provided in follow-up studies with sonorants, with the voice onset time of stops, and fricative consonants. So what I want to stress out with this study, with these studies, is that a single vowel and consonant, which is a very small segment of speech, can convey at the same time substantial multimodal information, which we can employ to understand language pathologies in patients and determine relationships between brain damage and speech production. For example, a vowel sound, which is just a few milliseconds long, conveys information about the sound, whether it is a vowel or a consonant, information about vowel quality, whether it is an E vowel or an A vowel, about the dialect of the speaker, whether the vowel is stressed or unstressed, whether it is produced by a typical speaker or an atypical speaker. It also indicates the gender of the speaker and the age of the speaker. So the multimodality of information provided by a single sound is impressive. And it is also suggesting for the use of acoustic features for the evaluation of patients. Just like speech acoustics, morphosyntax can provide substantial information to distinguish patients with different PPA variants. Natural language processing provides a very mature framework for analyzing and labeling text transcripts of speech conversations into grammatical constituents. Words are labeled with the corresponding parts of speech category, like nouns, verbs, adjectives, etc., using part of speech taggers. It also provides frameworks for identifying the relationships between words or the morphological categories of these words. namely their parts of speech. Constituency parsing and dependency parsing are two linguistic approaches that describe the organization of the syntactic constituents into hierarchical domains. These systems are very accurate. For example, recent constituent parsers now exceed the 90% accuracy on evaluation corpora, such as the Penn tree bank. Basically, often they perform much better than humans. To show the contribution of morphosyntax to differentiate PPA variants, we analyzed connected speech productions from 52 patients with PPA, 19 with the non-fluent PPA variant, 18 with the logobinic PPA variant, and 15 with semantic PPA. The transcripts were analyzed morphosyntactically with a morphological tagger, a natural language processing algorithm that provided automatically the parts of speech category for all words produced by patients. From the tag corpus, we elicited both content words, namely the proportion of nouns, verbs, adjectives, adverbs, and function words, namely conjunctions, prepositions, determiners, modal verbs, etc. And also the ratio of nouns and verbs with other parts of speech, like the noun-verb ratio, the noun-adjective ratio, the verb-adverb ratio, etc. The analysis of connected speech productions enable us to draw substantial observations about the part of speech patterns of patients with primary progressive aphasia. Some of these were expected, but some were unexpected. It showed that patients with the non-fluent PPA variant produce more content words than function words. See the top left figure for the content words and the top right figure for the function words. The content function word ratio is shown below. Patients with the non-fluent PPA variant produced overall fewer grammatical words compared to patients with the logobinic PPA variant and the semantic PPA variant. Nouns, shown in the upper right figure, were more frequent in patients with the non-fluent PPA variant than in patients with logobinic PPA and semantic PPA. This finding corroborates earlier studies that suggest naming is not as impaired in patients with non-fluent PPA as in patients with the semantic PPA. For example, see the study by Bastianse and Jongers and Cornodempini and others in 2011. We found that patients with semantic PPA variant produced fewer nouns than patients with the non-fluent PPA variant and the logobinic variant, which aligns with our expectations that these patients with semantic PPA variant are characterized by impaired naming abilities, which influences noun production. Verb production is shown in the bottom right figure. We didn't find significant differences between patients with different variants. Given the central role of verbs in sentence structure, it was surprising that patients with non-fluent PPA didn't differ significantly in the production of verbs compared to patients with the semantic PPA and the logobinic PPA. But that was also observed in other studies like Fraser 2014. This suggests that verb production might not be a very strong predictor for PPA variant classification. Patients with the semantic PPA variant And the logobinic variant didn't differ in the production of adjectives, shown in the top left figure. Patients with the semantic PPA produced more adverbs than patients with the non-fluent PPA and the logobinic PPA variant. See figures top right and bottom left and right for the noun adverb ratio and verb adverb ratio. Adverbs modify the sentence and or the verb phrase by providing information such as about the manner, place, time, frequency, degree, and so forth. The proportion of adverbs, the noun adverb ratio, and the verb adverb ratio were found to be important in distinguishing individuals with a semantic PPA variant from healthy controls, but none from non-fluent PPA variant. By comparing these productions to typical speakers, it is possible to score the grammar of these patients. So, automatically elicited acoustic and morphosyntactic measures can distinguish patients with mild cognitive impairment from healthy controls. They can also show differences between patients with different variants of primary progressive aphasia and estimate treatment efficacy. In the following, we will discuss how these acoustic and morphosyntactic measures can improve the diagnosis and subtyping of patients. Now we will be using machine learning for the classification tasks. So what is machine learning? I will provide an informal definition. Machine learning is a domain of artificial intelligence that enables systems to automatically learn and improve from experience without being explicitly programmed. Machine learning is fed data by the human expert, the clinician, or the doctor, and provides an informed output to the human expert. In general, machine learning models learn from data, and as they are trained on new data, their accuracy increases. Machine learning employs different types of learning. The most common ones are the supervised, unsupervised, and reinforcement learning. The supervised learning is a method of learning where the model makes associations between a target variable and a set of input features. Subsequently, the predictions of the model are evaluated on unknown data. So, for instance, if we want to identify whether a person is a man or a woman, we train the model on data that differentiate, say, men and women. For instance, the height, the weight, etc. And then we provide these figures again to see if by having only the features, the model can make the correct prediction about the gender, namely to say whether a person is a woman or a man. The unsupervised learning is a method of learning where the model identifies structure in the acoustic data, and usually here the data are unlabeled. Namely, we don't say what we expect the model to find, just to find the best grouping of the data. And reinforcement learning is a method of learning incrementally by adjusting the model. A learned behavior is accompanied with positive feedback or reward, whereas a behavior that is to be avoided is accompanied by negative feedback. So we can use machine learning first to augment existing tools' accuracy, such as the mini mental state exam. The use of automatically elicited morphosyntactic features in combination with machine learning can increase the reliability and the evaluation outcomes of existing tasks, such as the MMC, a screening tool for cognitive impairment. This test has been extensively validated and is widely used, but the MMC has also been criticized as not being effective in determining mild cognitive impairment. In a study by Katie Fraser and others, we examined the utility of augmenting MMC scores with automatically extracted linguistic information from a narrative speech task to better differentiate between individuals with mild cognitive impairment and healthy controls in a Swedish population. The machine learning model improved with the addition of just 4 linguistic features, morphosyntactic features, namely the AUC score, which measures a trade-off between sensitivity and specificity shown in the figure, is improved from 68% to 87%, almost 90% in a logistic regression classification. In a subsequent follow-up study, we employed only acoustic measures from speech fluency and prosody, and we found that just 1 acoustic measure can improve the AUC score from 77% classification to an almost 89–90% classification when the model adds acoustic features to the MMC score in a boosting classification algorithm. These results suggest that the accuracy of traditional screening tools may be improved through the addition of computerized language analysis. Now, we can also employ acoustic and morphosyntactic features to make a diagnosis of patients to identify patients with mild cognitive impairment from healthy controls. In this study, we employed neural networks. So neural networks are the state of the art in machine learning. There are different types of architectures of neural networks. For example, a feed-forward neural network is a sequence of layers with an input and an output layer, and several hidden layers. The input gets the data, for example, the morphosyntactic and acoustic features, etc. And the output layer provides performs a classification. For example, it will tell us whether a patient is a patient with mild cognitive impairment or is a healthy control. The convolutional network and the autoencoders are types of feed-forward networks. The convolutional networks are good for image classification, for image recognition, and autoencoders are employed in dimensionality reduction problems. Recurrent neural networks, such as the LSTM, are good for learning data such as text and sounds, which have a sequential structure nature. So they are employed in speech recognition, for example. More recent algorithms, such as BERT and similar models, these are successful to understand language by understanding long-distance relationships between, such as anaphora and cataphora. Anaphora is like when you for instance, when you make an association with a pronoun to a preceding noun. Mary, and her dress. Her refers to Mary. And cadaphora, we can say here, she who spoke at the conference is Mary. So we refer basically back to the name. The models I will be presenting are based on feed-forward neural network architectures. The term deep neural network I am using here means that the network has additional hidden layers. So although to this day there is no cure for dementia, early stage treatment can delay the progression of patients with mild cognitive impairment. Thus, the development of valid tools for identifying early cognitive changes is of great importance. Deficits in speech communication performance constitute a symptom in these patients, although memory and difficulties in remembering events and situations, along with problems in decision making, planning, and finding their way in familiar environments are the most prominent ones. We developed an automated machine learning method using a deep neural network architecture that targeted the linguistic deficits in speech production to identify patients with mild cognitive impairment. To classify these patients from healthy controls, we collected speech materials from the cookie theft picture descriptions during evaluation sessions as part of the Gothenburg MCI research study. 30 healthy controls and 25 patients with mild cognitive impairment, all between 55 and 79 years old, participated in the study. Their recordings were automatically transcribed and segmented. The segmentation was semi-automated. See an example in the figure at the bottom left. Measures of vowel duration and vowel formants from formant F1 to F5 and the fundamental frequency were calculated from the speech signals of vowels. Specifically, we provided an input as input 24 acoustic and sociophonetic predictors, namely the first form frequencies, fundamental frequency, vowel duration, and participant gender and age. To learn the acoustic characteristics associated with mild cognitive impairment in healthy controls, I trained and evaluated 10 feed-forward neural networks in a 5-fold cross-validation task, and by splitting the data into 90% training set and 10% evaluation set. The findings suggest that first, that the acoustic features provide substantial information for the identification of patients with mild cognitive impairment. The deep neural network architectures can classify patients with mild cognitive impairment and healthy controls with high classification accuracy, namely 83% classification from just 1 minute of conversation. And the deep neural network architecture proposed here can also contribute to the early diagnosis of cognitive decline, and we can quantify the progression of the condition using this same method over time. So a second line of research, which is related to this, is to develop fully automated end-to-end systems that can identify the variant of a patient with primary progressive aphasia from a single speech recording. Okay, primary progressive aphasia is a progressive neurological condition that is characterized by gradual deterioration of speech and language So we need basically to understand their acoustic production, their speech production, but we'll also need to understand and compare the grammar between patients to make the classification. Previously, we have seen that the grammar, that morphosyntactic features can distinguish between patients with different PPA variant. So remember the sounds we played at the beginning. The patients with the non-fluent primary progressive aphasia are characterized by sparsity of function words and abnormal syntax. Patients with the logopenic, with the semantic, patients with the semantic PPA variant have difficulties in confrontation, naming, and single word comprehension. And patients with the logopenic PPA variant have problems like in single word retrieval in spontaneous speech, naming. They do lots of phonological errors, etc. So
there is a
so to address these issues, we developed an end-to-end architecture that starts by analyzing all the acoustic features, and then after it calculates the acoustic and morphosyntactic features, it sends these features to the classifier, which is again a neural network, to predict the PPA variant. So to analyze language and speech productions, we recorded 9 patients with the semantic PPA variant, 16 patients with logopenic PPA variant, and 19 patients with the non-fluent PPA variant in the cookie theft picture description task. The recordings were produced, processed acoustically and morphosyntactically using 3 different but interconnected computational pipelines. The 3 pipelines extract data which are then fed into the neural network. Okay, that I will show you later in the next slide. Specifically, the first pipeline processed the sounds and provided an acoustic transcription and segmentation of sounds into vowels and consonants. The sounds were processed using Themis and a Python program that we developed to provide a text file with the audio transcription of each word and segments the audio into vowels and consonants. The second pipeline takes the input from the first pipeline and extracts acoustic information from the segmented vowels and consonants, such as vowel formants, vowel duration, the fundamental frequency, and measures of voice quality. The third pipeline processes the transcripts from the first pipeline morphosyntactically and provides measures of characters, words, characters per word, the proportion of each part of speech, the ratio between different parts of speech categories like the noun-verb ratio, the noun-adjective-verb ratio, which we have seen in the previous discussion, that they provide good classification between these patients. Then the deep neural network was trained on all of these measures. Okay, we provided these measures, and also the training happens like this. The network has a set of measures about grammar and speech acoustics, etc. And then we tell the network that these measures belong to a patient with the non-fluent PPA variant. We do the same again and with new measures, and we tell the network that these measures belong to a patient with the semantic PPA variant or the logopenic PPA variant, for instance. Okay, as the neural network gets trained, it learns to associate measures with the PPA variant. Okay, So when we provide the new measures from an unknown patient, the neural network will make the best prediction about the PPA variant based on what the neural network had already learned, okay, during the training phase. We test this neural network and compare it to other 3 machine learning models, a random forest, a support vector machine, and then decision tree. I will not get into details with these models, but just to know these are highly established machine learning models. Let me change slide. So the DNN, the deep neural network, is shown in the figure in the right hand of the in the right part of this slide. It consists of an input layer with 350 interconnected units, followed by 8 hidden layers, and an output layer with a softmax activation, which is a type of activation that enables enables the network to make a multi-class classification. All machine learning models were then trained and evaluated using an 8-fold cross-validation method. Overall, the deep neural network provided 80% classification accuracy and outperformed the other 3 machine learning models. The deep neural network provided improved identification of patients with the logobinic PPA variant, the non-fluent PPA variant with respect to the semantic PPA variant. Patients with the logobinic PPA variant were identified 90% correctly, whereas only 5% of them were misidentified as non-fluent PPA. Patients with the semantic PPA were correctly identified in the 65% of the cases. 30% of these patients were misclassified as logobinics, and 6% of the and 6% as non-fluent. 90% of the patients were correctly identified as non-fluent PPA, where only just 10% of them were misclassified as semantic. Note that we also asked 3 trained speech-language pathologists who didn't work with the patients to perform a similar task, namely to listen to the cookie theft samples from the patients that were used for the evaluation of the model and provide an estimation of the PPA variant. What we found out was that the 3 clinicians display substantial variation in their classification scores, with a mean classification accuracy close to 67%. So using the same evaluation data, the deep neural network provided higher accuracy in the classification tasks than the SRPs, and at a much faster rate. So the manual subtyping of patients with primary progressive aphasia is time-consuming and requires substantial expertise on the classification criteria, costly scans, and lengthy evaluations. It also requires substantial training on PPA variant classification. With this work, we show that the process can be automated with a combination of neural networks and natural language processing algorithms. As automatic assessment is typically quick and easy, it can enable multiple assessments and evaluations of patients in a certain period, being at the same time less stressful for patients. So, what is the way forward? The developments in machine learning and natural language processing are exciting. These methods will complement traditional methods in the clinic And for this reason, machine learning, artificial intelligence, and natural language processing are high in the priorities of the NIH and other funding institutions, setting the goals for active research in the field. However, we need also to take measures to make the best out of these models. We need to collect flagship data generalization. We need more data for predicting patient outcomes from longitudinal studies, for example, and elicit data from different populations. We need open data policies and flexible data access policy, reasonable and a reasonable data access process. We need also to employ data that scale up to large and diverse populations. Multilingualism and social aspects are part of the standard population and also part of our patients. So it's good to take multilingualism into account. Multimodal data are also important to be collected, namely consider variable modalities of speech communication, such as vision, sound, etc. Data from wearable devices, biomarkers, in addition to other data from speech conversation. For example, in the figure, we see that it is possible to use machine learning techniques to associate words, to associate objects with images. And we can also basically use similar techniques for explaining speech conversation. Finally, we need to evaluate different state-of-the-art machine learning algorithms, that is neural networks such as feed-forward, convolutional, LSTM, bidirectional neural networks like BERT, etc. And also use the current technology like websites, our telephones. To this, and I'm closing, just would like to present Themis, which is my way to contribute to this research. Is a website that enables the automatic transcription of sounds. The speech-to-text system is an ongoing technology, of course, and it is based on machine learning. It also this website enables us to analyze, to get some basic acoustic measures, such as voice quality measures and prosodic measures that I discussed. It can also allow us to elicit measures of the morphosyntax and the type token ratio, for example, and several others, which I'm adding basically continuously. And so this is basically a work in progress, and I hope we will be able to release this relatively soon. With this, I would like to thank the volunteers that participated in the clinical trials and their families for their selfless dedication to scientific progress. And I want to thank all of you for listening today.
Thank you very much, Harris. Okay, we have some time for questions, and the questions are coming in to the chat box. I started typing a few, but let's start with other questions from the audience. Manuel Mate asks, what is the extent of human input required for pipeline 1? So audio, text, transcription, and segmentation. How much human input is required?
So in that specific model, we try to avoid using manual analysis. So we're trying to develop an end-to-end system that gets the input and provides an output. So in all three PPA, so in all three pipelines, we don't have a manual evaluation. And what the thing is doing basically is that it allows us to basically evaluate the results of the model by doing everything automatically. And we can see that just by having this automatic approach, okay, we can get a very high classification output. Basically, the way we evaluate the three PPA pipelines is by seeing what happens with the model. What is the accuracy of the model? Can the model predict the patients with different PPA variants? And we see that it has an almost 83% classification accuracy. So it's doing well by classifying PPA variants. Why I'm saying that? Because we need to think that the subtyping process is even more difficult than the diagnosis, because the subtyping process will basically group within the same population of patients. Okay, so that was our goal in that paper, to avoid having manual effects on the pipelines and to see how well we can build a model that starts from the sound and provides an output.
Thank you very much. Related question from Alex Swiderski. Given that you are automating transcriptions, can you talk a little about the accuracy of the algorithm doing this? So the accuracy of the transcription itself.
Yeah, so the first thing that I have to say about the transcription part is that it is not a static model. The model that I used like 2 years ago and the model that is still in development now provides, has different, makes, say, different decisions. At the time, we had very few missed words, in the sense that it was close to 85% accuracy, if I remember well, because that paper was from kind of a year ago. Yeah.
Okay. And again, I think a related question, which was for me. Can your current model match the diagnostic accuracy of an average speech pathologist? So not someone who's an expert in PPA, for example, but you know, a regular speech pathologist in the field working on different types of disorders. And if so, how, if that's possible, how large of a speech sample suffices, you think, to reach that level of accuracy?
Absolutely. First, we did some trials with 3 speech and language pathologists, and we asked them, can you listen to the sound and can you make a prediction of the PPA variant? Some of the speech pathologists were really good. They had 1 or 2 misses, but some didn't do so well. So the variation of the speech between the population of the speech pathologists is great. So their assessments vary. Now, though, that you are saying this, it is important to evaluate the models based on the larger population of speech and language pathologists to see how well these models can help them and do an assessment if they are not experts, say, in a specific population, like patients with primary progressive aphasia. And the second thing is about the data, how many data we need. If you ask me, the potentials of the machine learning model is tremendous. We lack data. The more we collaborate, the more we build collaborations between universities, between hospitals, to collect data and train algorithms, these algorithms will improve substantially. And what is actually behind the current advances in machine learning is the ability to collect lots of data in several fields. For example, we see now with Tesla having autopilot in the streets and is doing very well. The same is with our machine learning models. So they can do very well and much better than they are doing now when we elicit much more data.
Okay, thanks. Yeah. Another thing I was just wondering about is, so in PPA, the way you're presenting that study, there is, of course, a level, I would suggest, of circularity, right? Where we diagnose the different types of PPA based on the behavior, typically, and then based on that same behavior, your own automated system also works. So I was wondering if you've tried to run an automated speech analysis predicting the different etiologies, so the histopathological basis, rather than the behavioral patterns.
Exactly. Yeah, no, we didn't do that at this point, but we are working on it. With this population, we only have these associations, but it is things that we are discussing in the lab, and we will be starting working on a similar project. Yeah. Mm-hmm.
Cool, thanks. And I have a question from Natalie Gilmore. Thank you for the talk. Do you see Themis being a tool that would be widely available to use for discourse transcription for people with aphasia, which could then be used for assessing treatment outcomes, in addition to the benefits you stated related to diagnosis, predicting treatment outcomes, subtyping? What do you need to make that possible?
I hope so. That the idea is to help the clinicians automate several tasks. For example, even narrow tasks, in the sense that if someone has his or her own transcriptions, they can put them into the system and get say an Excel file with information about the morphosyntax, the semantic. The use of Themis is really easy, in the sense what you do is basically copy paste the text and get an output, or upload the speech sound and get an output with the acoustic features. You don't need to do anything else. You don't need to go and do much of the pre-processing. So it will work well with when we have like an interest on generic patterns. Okay, if you want to do something very specific, I guess you will need the very specific tools for doing the specific task. But for most of the usages, I think Themis will be okay. Mm-hmm.
Yeah, and I think so. The last suggestion that Natalie makes with the question, what do you need to make that possible, is contributions of study data, right? That's I think you've already mentioned
exactly.
That.
I believe that we need more data, much more data. I don't know how much I have to stress that. This is very important to have access to good data that will enable us to do several tasks, not just simple diagnosis and subtyping. It can enable us to do automated prognosis if we like data over time, for instance. And yeah.
Excellent. Well, I think people will be able to reach you, Harris, with all their study data based on this talk and on the YouTube recording or the recording that will be placed on YouTube. So you can look forward to lots of emails, I'm sure.
Absolutely.
Thank you so much. We've come to the end of our questions. Thanks so much for your talk. Thanks to the audience for attending. And remember, in 2 weeks' time, we'll be listening to Rolien Bastiaanse. So we hope to see you back.
Thank you, Dirk.
Bye-bye.
Thank you all.
Happy Thanksgiving.
Happy Thanksgiving.